Hi everyone, my name is Natasha Caminsky and I'm a master's student at the University of Western Ontario in London, Canada. This is my colleague, Eliseos Mukaki, and my supervisor, Dr. Peter Rogan.
We are the authors of a review article recently published in F1000 Research. Our work summarizes all of the literature on information theory-based mRNA splicing mutation analysis.
In this video, Natasha will describe the highlights of our paper.
Our goal is to help you better understand information theory-based analysis, give you an idea of the conclusions that we drew from our review, and help you learn how to apply our methods to your own work. So before we begin, here's the full title of our article. It is part of the Rare Diseases collection of F1000 Research. Alright, let's get started. Our lab is specifically focused on improving ways to understand how sequence changes affect, in particular, mRNA splicing. Splicing is the process of recognizing the exon-intron boundaries and cutting, or splicing, out the introns. Defective splicing can have a number of different consequences, and depending on the mutation context, that can affect gene expression and/or function, and thus result in a deleterious phenotype. Over a decade ago, our lab created an online tool, the Automated Splice Site Analysis Server, based on Shannon information theory. This tool can be used to analyze a mutation associated with virtually any disease or condition. Over the years, it has been used in over 200 articles to analyze over 1,700 variants in all sorts of rare and common diseases. For your interest, the different sections of the review are listed here. You can pause the video if you want to take a longer look. If you are not familiar with information theory, I strongly suggest that you take a look at those sections of our paper because I'm only going to briefly summarize it here. Information theory, or IT, quantifies information in bits and can be applied to almost any discipline that involves communication. It's the reason we're able to use our cell phones to carry on multiple conversations over the same network at the same time. Known donor and acceptor sites across the genome are used to create these sequence logos that we see here on this slide. When aligned, the frequencies of each base, A, C, G, T, at each position can be converted to their average information contents, which have a maximum of 2 bits at any position. You can then compute a sequence logo, which visually represents the frequencies and the information as a stack of nucleotides, whose heights are proportional to the relative frequencies of the bases at each position. However, contribution of each sequence to the average information can also be determined. This is called the individual information of the sequence, or its RI value. To determine the consequence of a variant on these sequences, all you need to do is compare the information of the wild-type sequence, which is termed RI initial, to the information of the variant sequence, termed RI final. The change in information, which we call delta RI, is a measure of the severity of the mutation. For many polymorphisms, delta RI is negligible. A negative RI value is nearly always a mutation because of the relationship between information theory and the second law of thermodynamics, which shows that the change in information is directly related to the thermodynamic entropy and free energy of binding of a sequence. I'm now going to go through our paper in order to get the main points across. This graph represents the distribution of deleterious natural site variants that we came across in our literature review. In total, we found 431 affecting acceptors and 604 affecting donors. As you can see by this figure, the frequency of deleterious mutations reported in the literature is highly correlated with the average information content at that position shown in the top panel by the sequence logos. This does not mean that more mutations occur at these positions than at others, but rather that the susceptibility of a particular nucleotide to mutation is directly related to the information content at that position. The next figure is a screenshot of a program we developed called Splicing Mutation Calculator, which I'll now do a quick demonstration of. This software provides a compact way of looking at the published evidence to date for mutations that alter the information contents of constitutive splice sites and benign polymorphisms that do not have a significant impact. Before you begin, you need to know which variant you want to analyze. In this example, I have put the variant that I want to analyze in the top right corner of the screen. The first step is to then select the type of natural site in which it occurs, which means that in this case I'm going to choose the acceptor site. Next, I need to select the position using the slider bar, so I'll do that. Then you need to select the wild type and variant nucleotides, so A and T. Then you can click Submit. A table will appear indicating the variant, the number of times that the variant was documented in the literature that we reviewed, and the computed change in information content using both the old and new information models. These differ primarily by the number and composition of the splice sites used to compute them. The changes in information are color-coded to indicate the predicted effect of the mutation, whether it be deleterious, probably deleterious, leaky, or benign. Finally, by clicking on View Above Variants in Source Table, it is possible to view more details about the variants reported at this position in the literature. In this figure, we show that many splicing mutations do not simply result in skipping of an exon, and alternate transcript structures are sometimes the predominant consequence of splicing mutations. In fact, some cryptic splicing mutations do not involve the constitutive splice sites at all. This figure illustrates the ways in which cryptic splicing can be activated. We have also indicated the number of cases in the published literature that reported these types of activation. In parentheses, we have indicated the percentage of cases that were correctly predicted by information theory analysis. This is the last figure in our paper. We plotted the local distribution of activated cryptic sites relative to the natural sites of the same polarity to discern any trends that might be useful in anticipating where such sites might be concentrated. The bottom panel is a magnification of the region circumscribing the natural site. We actually found that there is no preference for observing activated cryptic sites in either the adjacent intron or exon, and also no preference for activation in a donor versus an acceptor. In fact, activated cryptic sites occurred over a wide range of distances both up and downstream of the natural site. This review of the literature showed us that information theory-based splice site analysis has been effective in numerous studies at predicting the impact of a sequence variant on splicing. A careful analysis of the studies we reviewed also allowed us to identify misinterpretations that are commonly made when using information theory analysis, as well as some limitations of the methods. So we have come up with general guidelines for our users. First, we advise that the gene isoform and genomic coordinate of the variant being reported be provided. This allows for easily reproducible analysis. Second, we recommend that RI values be reported. This gives the reader a more complete understanding of the impact of the variant analyzed. Third, because mutations within the exon are known to cause cryptic splice site activation, it is important to analyze coding sequences and not dismiss the potential effect a missense or synonymous mutation may have on splicing. Fourth, if possible, it is advisable to conduct functional validation of potentially deleterious variants that have been identified. Fifth, we suggest that the coordinates or the size of sequence window used to conduct the in silico analysis be included in the description of the analysis. Should the window not be wide enough, it is possible that potential cryptic sites will be missed. Finally, when analyzing genomic variants other than single nucleotide changes, we recommend that the genomic coordinate entry method be used in order to ensure the proper parsing of the variant. To learn more about information theory, you can take a look at some of these articles. To better understand the different information theory using tools that we have developed, check out these articles. Thank you for taking the time to watch this video. We hope that you find our article useful for your research or diagnostic analyses. If you have any questions, please don't hesitate to contact us at the correspondence address in the paper or the ones that you see on the screen. Remember, no matter which mutation is present in whatever gene, our tools can provide insight into interpreting its impact on splicing.